So we're going to continue our discussion of Chapter 2 of our biochemical basis of human biology by looking at the actual molecules that are going to have function in the human body, in addition to the things that we've already looked at. So when we tend to look at these organic compounds, these molecules of life, a lot of times we give these the moniker macromolecules because they are very large. They fall into four main classes: the carbohydrates, the lipids, the proteins, and the nucleic acids. And these are like the long polymer chain versions of these. And each of them is made up of single subunits called monomers. And so we can look at the subunits of each of these. So carbohydrates are made up of single sugar subunits, like glucose and fructose and galactose. Lipids are made up of fatty acids. Proteins are made up of amino acids. And nucleic acids are made up of something called nucleotides. And so this is kind of an outline of the rest of this lecture. We're going to go through each of these organic compounds and talk about how they're put together from these subunits and broken down into these subunits. And we're also going to talk about one called ATP, which is also another important big molecule. Now there's two main chemical reactions that are very important in this discussion. There's one chemical reaction that each of these use to build up the polymers, to build up the big long chains from these individual monomers. And that is called dehydration synthesis. Synthesis means to put things together. Something that's synthetic has been synthesized. When we synthesize things, we put it together or build it up or make it. The other chemical reaction, all of these polymers use to break down into the monomers, and that's called hydrolysis. So there are 8 types of molecules that are on this screen right now, and you only need to learn 2 chemical reactions to understand how all of these behave. So when we're talking about chemical reactions, a chemical reaction occurs when some reactant, some molecule changes in some way. And we write these as chemical equations. What goes into the reaction is called the reactant. What comes out is called the product. And if there is an enzyme that catalyzes this reaction, the name of the enzyme is written above the arrow. The enzyme isn't really a reactant and it isn't really a product, but it participates in that chemical reaction. And then the 2 that we're going to talk about are dehydration synthesis and hydrolysis. So, for example, one that we use pretty commonly when we're learning in this chapter is the idea that there is a sugar called sucrose. Sucrose is a disaccharide. It's made of 2 sugar subunits. It's made up of glucose and fructose. So when sucrose is hydrolyzed or broken apart, it's hydrolyzed into glucose and fructose. Now there is an enzyme that catalyzes this reaction, and that enzyme is named sucrase. We're going to talk about that as our example. That can be found, there's an animation that can be found in the Learning Materials tab that shows how sucrose takes place, how sucrose hydrolysis takes place with the sucrase enzyme. So your book shows you thinks it's on this page. Okay, sorry about that. Your book shows you a figure that is an example of both dehydration synthesis and hydrolysis. And I find that the more simply that we can get into this discussion, the easier it is to start to understand. And so I like this figure because it doesn't show you any part of the molecule except the part that is important. So we have this short polymer. It has 1, 2, 3 monomers joined together. So it's this short macromolecule. And we're going to grow that macromolecule. We're going to make it bigger and longer, a longer polymer. And so the only part of this whole molecule that's important is this hydrogen that hangs off the side. And then here is one single monomer. This could be a glucose or fructose or galactose. It could be an amino acid. It could be a fatty acid. It could be a nucleotide. It could be any of our individual subunits that we're attaching to this carbohydrate or this protein or this DNA molecule. And the only part of it that's really important is that it usually has an OH hanging off of it. Because you can see that this H and this OH form water. Water comes out. That's exactly what dehydration means. And in its place, we form a bond, a new covalent bond to form our longer polymer. So this is called dehydration synthesis because water comes out and we form a new covalent bond to make this longer polymer. Dehydration synthesis joins a small polymer and a single subunit or a bunch of small single subunits together to form a longer polymer. So if we're writing this as a chemical equation, we would write our short polymer and a single subunit. Those are what go into the reaction. Those are our reactants. The arrow tells us that a reaction has taken place, and then a longer polymer is our product. What's an example of this reaction? Well, I'm going to give you an example that's going to make a little more sense in a few weeks when we talk a little bit more about cell function. When we're making proteins, we start out with a short polypeptide, and we add to that amino acids. And we form what's called a peptide bond. And that forms a longer protein molecule. That's catalyzed by enzymes and proteins and RNA molecules that are found on the ribosome. And so once we go through cell structure and cell function, I'm going to have you come back and look at this part of the lecture because I bet this is going to make a lot more sense. But that's one example. We're starting to put together amino acids and proteins to make a longer polypeptide chain, to make a bigger protein, a larger macromolecule. Now, hydrolysis is just the opposite. Hydrolysis adds water. Hydrolysis literally means water breaks apart. And so water breaks this bond. What happens to the water? Well, the H part of it goes here, and the OH part of it goes here. Now, if you look really closely at this figure, this is the exact opposite of the dehydration synthesis. We put these two together by dehydration synthesis. Dehydration synthesis. That's the opposite of hydrolysis. Hydrolysis breaks them apart, and dehydration synthesis puts them together. So one example is the one that we've already looked at, sucrose being broken down into monomers like glucose and fructose. And here we use the enzyme sucrase to do that. So now we can take those two reactions and we can apply them to all of these individual states, all these individual sets of macromolecules. So we can apply dehydration synthesis and hydrolysis to carbohydrates, into lipids, into proteins, and nucleic acids, as we talk about each of those macromolecules in a little bit more detail. So carbohydrates are called carbohydrates because they contain carbon and hydrogen in equal amounts. Sorry, carbon and water in equal amounts. So the basic formula is however many C's you have, that's how many H2O's you have. The way humans use carbohydrates is as an energy source. There's a whole process by which we take the electrons that are in these atoms and we use it to form something called ATP, which is a molecule that has a lot of energy stored in it. And then we can use ATP for cell function and cell work. We have monosaccharides, which is one single carbohydrate subunit. If you put two of them together like sucrose, that's called a disaccharide. Once we get past that, we just use poly, which means many. And so disaccharides and polysaccharides form from those monosaccharides by dehydration synthesis. You don't have to memorize any structures. I really just show you this because it goes along with the previous figure that I showed you. Now instead of those purple balls, we're actually showing you some structure of some of these sugar subunits. But they all have an H on one end and an OH on the other end. So they can all go through dehydration synthesis and hydrolysis. If we combine two of these subunits by dehydration synthesis, it shows you in this figure where the water is formed. And in lieu of that, we form a bond, and this has formed sucrose. We form this by dehydration synthesis, and we break it down by hydrolysis. Those two reactions are the exact opposite of one another. We can build up to even larger and larger and larger chains of polysaccharides. What they're showing you here is glycogen, and this is really important in human physiology. This is how glucose is stored in the liver and also the skeletal muscles. It's these long branched chains of glucose, and to get glucose out of the liver, you just break these bonds, and that releases a glucose molecule, a glucose monomer. We have two hormones that control this process that we're going to talk about. One is the hormone glucagon. Glucagon causes the release of glucose from glycogen in the liver. So when blood sugar levels fall, the liver hydrolyzes that glycogen to yield glucose. The other hormone is insulin. When blood glucose levels are high, insulin facilitates the taking up of glucose into muscle in the liver for storage. Again, dehydration synthesis and hydrolysis are going to be the exact opposite reaction from one another. Lipids are a little bit unique. Of our four, they have very few polar covalent bonds. Most of their bonds are nonpolar. They have a lot of bonds between carbon and hydrogen. And carbon's not electronegative. It's one of the ones that was taught to share electrons. It was taught to play fair and to share electrons, and so it makes these very non-polar covalent bonds. That's very nice, except that that makes this very hydrophobic, because water is polar, and water is the solvent in most of the aqueous solutions in the human body. So lipids become mostly insoluble in these polar solvents, such as water, mostly insoluble in body fluids. To get it through the bloodstream, we have to combine it to transport proteins called lipoproteins to transport in the bloodstream. But lipids have a very specific function based on this. Phospholipids are going to help form our cell membranes, and they can form a really specific barrier because they're so hydrophobic. Now, all of these lipids, like triglycerides and phospholipids, are made up of fatty acids. Again, you don't have to memorize structures, but you see lots of carbon, hydrogen here. There's just a little bit of polarity at one end of the molecule. So the majority of this molecule is not going to dissolve in water. It's going to be very hydrophobic. That helps us to form this triglyceride by dehydration synthesis. Oh, look, there's the water that's formed. In the case of the phospholipid, we link those fatty acids to a very large molecule. It has a lot of phosphate groups, which are very polar. Phosphate is another very electronegative, like oxygen. So there's a lot of oxygen here. There's a lot of phosphate here. And so this blue bubble that they're showing you is often called the polar head of the phospholipid. And then these fatty acid tails are the part that are very hydrophobic. So we simplify this by drawing the polar head as kind of this round molecule, and then these little fatty acid tails. And so this is the hydrophilic head portion that faces the water, that faces the extracellular fluid and the intracellular fluid, whereas these fatty acid tails are very hydrophobic. And so they end up associating with each other in the center of the cell membrane. So in the next chapter, when we talk about cell structure and function, we'll come back to this mixed structure of the phospholipid because it creates a really great barrier for the cell membrane. The hydrophobic tails on the inside and the hydrophilic heads facing the extracellular fluid and the intracellular fluid. So now we're going to talk a little bit about proteins. Again, proteins are going to be put together and broken down the same way. But whereas we have probably 10 or 15 different fatty acids that we find, there's maybe 5 or 6 different sugars that we find, there's 23 different amino acids. And proteins are a lot longer polymers. So we can form a number of different protein structures that have function in the human body. And so proteins can do lots and lots of different things. Basically, carbohydrates provide energy. Lipids can provide energy and barriers. And DNA and RNA sends information. Everything else is done by proteins. So they form the structure of cells, tissues, organs. They form contractile proteins in lots of different muscle cell types that cause parts of the body to move. They form transport proteins, both to get things in and out of the cell, as well as to get things through the bloodstream to different parts of the body. They form buffering proteins to try to help maintain pH. They form antibodies and other components of the immune system to provide immunity. And almost every chemical reaction is going to be catalyzed by an enzyme. So they have very important function. And so we can talk a little bit about their structure. Dipeptides are going to be 2 amino acids. And then longer than that, we call them polypeptide chains. Now, up here, they show you the basic structure of the amino acid. It's designed around a central carbon. On one side, you have a carboxyl group, COOH. On the other end, you have an amino group, which is NH2. One of those bonds goes to a hydrogen, and the other goes to kind of the rest of the molecule. That's going to vary depending on which amino acid it is. So all that I've drawn, other than that R group, is the same for every single amino acid. And so you can see that there's an OH and an H hanging off each side. If we put 2 of these together, it'd be very easy to form a bond between them as we pull water out and do dehydration synthesis. Now these side chains, this R group, this part that varies with each amino acid, that can be as small as just a hydrogen. It can have a huge ring structure to it. It can be very long, or it can have a sulfhydryl group, and sulfur forms hydrogen bonds very easily, as does nitrogen. So these are going to behave in water very differently than shorter or polar side chains, or non-polar side chains. So when we start to think about amino acid side chains, there's a lot of ways that those can influence protein folding that we'll talk about in a few minutes. Their size can help determine where they show up in the folding. Whether or not their side chains are polar or non-polar can help determine where they show up in the folding. Whether they have any ring structures or sulfhydryls or anything else that's unique like that. So this is the figure that shows we put these 2 together, we form water, dehydration synthesis puts them together, hydrolysis takes them apart. Now in the case of the protein, the bond that's formed has a very specific name, and it's called a peptide bond. So once we string together those amino acids, proteins actually have to have some additional level of structure added to them. They actually have 4 steps of folding, or 4 levels of structure. The primary structure is just that amino acid sequence, getting the amino acids in the right order. Then you have some side chain interactions. So you start to have some hydrogen bonds. They can form either a little alpha helix, just a little helical structure, or this beta pleated sheet. But these are going to be localized twisting and folding of that amino acid chain. Finally, the whole thing is going to fold up in its tertiary structure, which is its 3-dimensional shape. And that's going to be stabilized by further hydrogen bond formation. Now, some polypeptide chains associate with others, and so that forms quaternary structure. And so I want to give you an example. Close the window, so I'm going to have to get into it. In the Blackboard course shell, So in the Learning Materials tab of your course, we have a whole folder on enzymes and protein folding animations. The particular one that I want to show you is this GCSF protein folding illustration movie. And so I'm going to play it through, and then I'm going to show you what it is that we're looking at. There's no sound. So if you don't hear sound, there's no sound to this video. So you're not missing anything. Okay, so it's not a very long video. So the name of this protein is the granulocyte colony stimulating factor. So this is a growth factor that helps granulocytes to grow. Those are white blood cells like neutrophils, eosinophils, and basophils. So this is a growth factor protein that's going to help cells grow. So what they first show you in through here is this primary structure of the protein. This is the amino acid sequence. And you can see some of the side chains have these ring structures that come off of them. Some are just very short, don't have much that comes off of the actual backbone chain, hardly at all. And they've color-coded it to give you the hint of where some of these secondary structures or localized folding is going to take place. Now there's 20 different amino acids. Each different amino acid has a one-letter abbreviation. So G, S, S, L, Q, L, L. L is probably lysine. I don't remember all of the names and the abbreviations. But what this part of the video is showing us is it's kind of taking out that extra side chain from each amino acid to show you what the actual primary sequence is by name. And then they're reducing it even further to just show you each amino acid as a little colored ball. So that when we first look at how the protein folding takes place, we're just looking at these basic levels of protein folding. So here's the primary level, the sequence of the amino acids. And then we start to form some secondary folding. We're forming some alpha helixes. This is an alpha helix. This is an alpha helix. In fact, each of the different colors forms a different alpha helix. Now we're starting to actually put all of these alpha helices together in the folded three-dimensional tertiary structure of this protein. They turn it around so that you can kind of see. Up here's where the active site is going to be. Then they go back and put in the letters of each of the amino acids from beginning to end, just like they did before. And now they reconvert it to the side chains. So they're reminding you that all these little balls are amino acids, and now they're showing you some of the side chains. And you can see that some of it's folded so that some of these ring structures kind of associate with each other. Some of these areas with very short side chains associate with each other. And then they give you a space filling model to actually show you what this protein would look like as an actual full molecule. And then it goes away. So I really like that video because it's really simple, but it's an actual protein. And so it kind of gives us all these steps that your book describes in an actual real life protein. Proteins have to be folded properly in order to have their function. So the three-dimensional shape of a protein determines its function. And if a protein becomes unfolded, we say it has been denatured and it will cease to function. The things that cause proteins to become denatured are things like changes in temperature, changes in salt concentration, and changes in pH. And guess what? Those are all things that disrupt hydrogen bonds. Because hydrogen bonds are those interactions between something that's slightly positively charged and something that's slightly negatively charged, if we add salt or if we add too much acid, that's going to change this interaction as it creates basically those hydration spheres that we looked at when we first studied those types of bonds. Now, one very specific type of protein is the enzyme. And enzymes are going to help chemical reactions to take place. That's going to be their function. They are proteins. So everything that we just learned about proteins applies to enzymes. They have to be folded properly. They go through those 4 levels of protein folding. And once they're folded properly, they can help speed up or facilitate chemical reactions. They help them go faster, or they help them happen in certain places. We can put an enzyme in a cell membrane to make the chemical reaction take place there. The reactant that binds to the enzyme is called the substrate. So the enzyme and the substrate come together and form the enzyme-substrate complex. The enzyme converts the substrate to the product, but the enzyme itself does not take place in the reaction. It's not a product and it's not a reactant. We would write it on the top of the arrow of a chemical reaction. And the enzyme can go on to further catalyze other reactions. And so this is a figure that shows that. So our substrates here are sucrose and water. Our enzyme is sucrase, and this blue part shows the active site of the enzyme. The enzyme and the substrate come together at the active site, forming that enzyme-substrate complex. The reaction takes place, and that sucrose gets broken apart into glucose and fructose. They get released, and the enzyme is unchanged and is ready to catalyze the same reaction again. And so it can go find more substrates. Why do we say water is a substrate for this enzyme? Because this is hydrolysis. This is sucrose breaks down into fructose and glucose by the action of the enzyme sucrase. And since this is hydrolysis, water breaks apart. Now, the bottom part of this figure gives you a space filling model very similar to what we looked at the end of that protein folding video. And here's the glucose, and here's where it fits into this specialized active site. The active site of an enzyme is extremely specific for a given substrate. So we can have other lactose is a sugar that is glucose plus galactose. The only thing different is one part of that molecule, but it's so different that it has a different enzyme. Lactase is the enzyme. So enzymes are proteins, and they help chemical reactions take place. Usually their names end in ASE or ZYME. They're going to be very specific and catalyze a particular reaction. They can catalyze the formation of a product from one or more substrates, but they're not changed in the course of the reaction. They can go on to catalyze other reactions. The important part is the active site of the enzyme. That's where the substrate binds. It needs to have its particular shape to function. And so changes in salt concentration or temperature or pH can change the folding of an enzyme and therefore change its function. Then our fourth kind of macromolecule is nucleic acids. We have two major types of nucleic acids, DNA and RNA. And they're made of nucleotides called pyrimidines and purines. These nucleotides have a structure we can look at. You don't have to memorize structures, but there's an OH hanging off one side and an H hanging off the other side so that we can put them together. And so what we do is we form this long backbone, and then we put two of these polymers together. And the way the two polymers are held together is by hydrogen bonds between parts of the bases. This is very similar to how proteins fold. You put together a chain of amino acids, and then those side chains do hydrogen bonding. Here we're putting together a string of nucleotides, and the parts that stick out are doing the hydrogen bonding. The other type of nucleic acid that we will talk about is RNA. The difference is the sugar that's involved in the nucleotide. DNA is double-stranded, whereas RNA functions as a single strand. DNA encodes the information for making proteins. And the other thing that's unique about RNA is there's three different types that we'll talk about. So all four of those macromolecules, if you go back to that original slide with that original list where we made that table, those are all polymers made up of individual subunits. We put together the polymer by dehydration synthesis, and we break it down into individual subunits by hydrolysis. Now, ATP is very similar in that it's formed by dehydration synthesis and broken down by hydrolysis. This is our molecule that we use to store energy. The way that we create ATP is by taking an adenosine diphosphate, and we add a phosphate, and we add energy. And that's going to form ATP and water, since that's a dehydration synthesis. And then we break down ATP by hydrolysis, giving off that energy that can be used for cell work. Now, I think that line about page 60 is from a different textbook. Yes, it is. The ATP to look at in your book, not even in chapter 2. If you look at page 39, it's showing you ATP being formed and broken down. And so what they show you is kind of a little bit of a representation. Adenosine has a part that's called adenine and a part that's called ribose. But what's important to us are these phosphates that are attached. If there's two phosphates, we call that adenosine diphosphate. If there's three phosphates, we call that adenosine triphosphate. And you see this covalent bond is actually denoted by a wavy line. That wavy line tells us that there can be energy stored in that chemical bond. So that takes us through all of the macromolecules, our 4 major categories. There's lots of molecules that fit into each of those categories. There are a lot of hormones that are either peptide hormones, that are proteins, or steroid hormones, so they're lipids. There are a lot of carbohydrates, long and short carbohydrate chains. And then there are a lot of DNA and RNA sequences that become important as we talk about gene expression and turning that DNA into a protein. So if you have questions about anything related to this lecture, please feel free to post it on the discussion board or send me an email.